Hello, everyone. Thank you so much for viewing my poster for the Discover presentation. Today, the topic of my poster for the Discover is investigation of photoactive metallodrugs for photodynamic inactivation of bacteria. In order to understand what's photodynamic inactivation of bacteria is, let me just briefly go over the mechanism. It's an alternate strategy in which a photosensitizer molecule is required, and this molecule, in the presence of light and oxygen, can produce cytotoxic reactive molecular species or reactive oxygen species, which are responsible for showing cell kill. So this was the basic mechanism behind photodynamic inactivation of bacteria. In our lab, we actually synthesize these photosensitizer molecules, which are either ruthenium-based or osmium-based coordination complexes, as shown here in the box at the bottom. My study for this poster is based on these ruthenium-containing BP molecules or BP ligands, which have CF3 groups attached as co-ligands as a modification. We pi-expand these molecules by the introduction of phenanthrolane, imidazole, and the thiophene groups into the molecule in order to stabilize the reactive oxygens, in order to stabilize the excited state species of these photosensitizer molecules. Because if you look at the mechanism, our molecule undergoes a whole energy diagram, schematics. Our ground state photosensitizer gets excited into the excited state in the singlet form, and after intersystem conversion gets converted into triplet excited state. This is the excited state which produces the raw species in order to return back to its ground state. So we're trying to stabilize the lifetime of this excited state of the molecule by expanding our photosensitizer molecule. As you can see that the molecule is pi expanding, but upon doing lipophilicity studies of our compound, we saw that there is a systematic change in the log D value, the distribution coefficient of the molecule as well. The negative value for shows that the molecule is more hydrophilic in nature, whereas a positive value shows that the molecule is more lipophilic in nature. And as you can see, there is a complete systematic change in the lipophilicity of the molecules, where our most hydrophilic molecules are the ones which are least pi expanded, and the highest pi expanded are more lipophilic in nature. Because we are depending upon, or we are saying that our mechanism produces reactive oxygen species, we need to know what's the energy, what's the lifetime of our excited state molecule. Are we increasing the lifetime of these molecules or not because we are pi expanding it? And also, how much singlet oxygen is being produced in turn in the excited state. So for that, we do photophysical characterization of our molecules in which we use lasers to understand or to find out the lifetime of our molecules. For example, I'm showing just a representative diagram of the excited state spectra of ruthenium molecule with 4-thiophene, like this one, with the 4-thiophenes attached here at the bottom. As you can see that we have a very excited state available between 500 and 700 nanometer, and the lifetime of this excited state molecule, the 4-thiophene, was actually increased up to 20 microsecond. Because we are talking about the stabilization of the excited state and also about the singlet oxygen in one of the mechanisms, we also measure the singlet oxygen needs for our molecules. This diagram here, this plot here shows the singlet oxygen yield change upon pi expansion of the molecule, where Rutris BTFMB is the least pi expanded molecule and 4-thiophenol is the most pi expanded molecule. And as you can see, the singlet oxygen yield increases and then it starts decreasing back, but it starts increasing again with the addition of 2 to 3 thiophenes, but with 4 thiophenes, it decreases again. We don't know the rationale behind this decrease, but there might be some secondary reactions happening due to which this singlet oxygen yield decreases for our 4-thiophene molecule. Next is the photobiological activity of our complexes. I test these compounds in both gram-positive and negative strains, namely E. faecalis strain, antibiotic resistant E. faecalis V587, and Abomani strain. And we also measure the pI values. We measure the activity under dark and light because these molecules are excited under the light conditions, so we need to confirm their activity. In order to know more about my study, please do stop by my poster during the Discover presentation. Thank you so much for listening to my talk.